Okay, so today I want to look at meiosis. So we've looked at mitosis and the cell cycle. The beginning bit of the cell cycle is identical, so we won't go over that again. So in other words, you have G1, S, G2, but at the end of G2, instead of mitosis, you've got meiosis and cytokinesis. So let's think about meiosis and what we know from GCSE. So we know that meiosis is used in the production of gametes. We know that meiosis involves 2 cell divisions, and at the end of the process of meiosis you end up with 4 cells, each of which are haploid. In other words, they have 1 from each pair of chromosomes, and each of which is genetically different to each other and to the parent cell you started with. So we're obviously going to have to look at what's happening in both stages, which we call Meiosis One and Meiosis Two, and how we introduce a genetic variation to ensure that they are all different. So if we go back and start thinking just about prophase, and we're calling it Prophase One because it's the prophase that was belonging to the first stage, the first meiotic division. So if you look at the cell in prophase, some of the things that occur in Prophase One we're used to. So for example, the nuclear membrane disappears. In addition, we know that the chromosomes condense. And they condense so that they can be seen as 2 sister chromatids connected by the centromere. The other thing that happens is that spindles start to form from the centrioles. So we'll put our spindle in. Just beginning to form from our centrioles, which are over there. Okay, but there is one very subtle difference, and that's the way in which the chromosomes behave. So we'll draw one set of chromosomes big and one set of chromosomes smaller. I'm going to draw them in different colours. That is deliberate. So what we notice is that the homologous pairs of chromosomes pair up. And when they pair up, they form these joins with one another. So if we have a look at this pair over here, okay, this pair is a pair of homologous chromosomes. So homologous meaning they basically have the same genes on them, not the same alleles, but the same genes on them. So both pair number 1 or pair number 2. So they pair up and they form what we call a bivalent pair. Now, let's look at what's happening at this bivalent pair. So let's take a little bit of a sort of picture of that. Let's go down and have a look. So we have 1 chromosome, and it seems to have formed a join with its pair over here, like that. And that join over there, that join is a chiasma, or plural is chiasmata. And really important, because what's going to happen at that join is they're going to swap information. They're going to swap alleles. So from this chromatid, they're going to swap information. So for example, it may be that originally this person was big A, small a, okay, and big B, small b. And remember, these 2 green ones are identical copies of each other. And what's going to happen is these 2 are going to literally switch. So you're going to end up with 1 chromatid that still has the big A big B allele, but the next chromatid is going to have the big A allele, but it's swapped information at the bottom. So it's going to end up with the little b allele over here. And on this side, you've got one chromatid with the small a, small b allele, but the other chromatid still got the small a, but now it has a big B. And all of a sudden, what we've done is from having identical copies of DNA, we had 2 identical copies there and 2 identical copies there, we've now ended up with 4 pieces of DNA, all of which are genetically different to each other. The idea being that one of each is going to end up in your final cell. So therefore, they're all going to be genetically different. And bear in mind that these chiasmata can go form many times along a piece of DNA. So you get lots of swapping. And we call this crossing over. Crossing over is when you switch alleles between chromatids that are on homologous chromosomes so that you create new combinations of alleles. So if we go back to prophase, this is what will happen. These will switch information and these will switch information. And this will become really important when we get onto metaphase next. So if we have a look at Metaphase One. So we have our cell, which was like that. We have our spindle, made of spindle fibers formed from the centriole. And the homologous pairs that we formed up there, those homologous pairs will now line up. They'll line up along the center. But remember that they have actually switched a little bit of information. So I'm going to draw them in like that with their switch, like so. Likewise with my blue ones, which are over here. Okay, so we have homologous chromosomes, homologous pairs line up on the equator. And essentially what's going to happen next is that this chromosome will go to this end, and this chromosome will go to this end, this chromosome will go to this end, and this one will go to this end. And this is really important because I've lined them up in a specific way so that basically the red and green will go off with the blue and pink. And the one which is mostly green with a little bit of red will go off with one which is mostly pink with a little bit of blue. But there's no reason, there's no logical reason why I couldn't have decided that actually that's not correct. And what I'm going to do, just redraw my spindle here, is I'm going to line it up the other way. I'm going to line it up with my one which is pink over here, and my one which is mostly blue down here. And if I do that, then I'm going to get a slightly different combination. My mostly red one's now going off with my mostly pink one, and my mostly green one is now going off with my mostly blue one. Now, the fact that they can line up in any orientation they like, and the fact that it can be done several times over, you've got 23 pairs of chromosomes here. This is called independent assortment. So independent assortment is the idea that they can line up in either orientation along the equator of the cell, and it actually doesn't matter, but it will affect the combinations that you produce, so therefore introduces variation. We then have Anaphase One. So in Anaphase One, spindle fibers will shorten, and when the spindle fibers shorten, they will now separate out those pairs of chromosomes. So those pairs that were up here will now be separated out completely. So I draw them in. This one, will go off separately from that one, and my one over here. Just going to check what colour I used in the end. So my pink one. Will go off over here. And we have separated our homologous pairs. So the spindle fibers contract. And homologous pairs are separated. We then get to telophase one. Now, telophase one does slightly vary depending on how long the cell is going to spend in meiosis one. But the idea of telophase one is that you've now ended up with two nuclear membranes forming, and you've got your chromosomes in here. Like that. Like so. Okay, and that's telophase one specifically. So you've got new nuclear membrane forming around just the chromosomes that have been taken to opposite ends. So we then move on to prophase one. Well, not much is going to happen in prophase one, unless it's been sat around in telophase for a very long time. Prophase two, sorry, unless it's been sat around in telophase one for a very long time. The main thing that's going to happen is you're going to start to form more spindle fibers, but they're going to form at right angles to the original spindle that was there. And you're also going to remove that nuclear membrane again. So we now have no nuclear membrane. That's gone. And that's pretty much it in prophase two. So what will happen in metaphase two? Well, metaphase two will seem to be very similar to something we've met before, because let's look at what happens in metaphase two. We have our spindles completely forming over here. And this time the chromosomes, they line up individually along the equator. Like so. Now, like before, it is up to them which way round they line up. So I've lined this one with the red one on that side and the red and green one on that side, but there's no reason that the chromosome couldn't line up the other way, which means we get even more variation, a second lot of independent assortments. And that's pretty much it for metaphase. The individual chromosomes line up along the equator, the same as they did in metaphase of mitosis. And then you have anaphase. So in anaphase two, Let's try that again. In Anaphase Two, the spindle fibers once again will contract. And this time when they contract, they're going to pull the sister chromatids apart. And when they pull the sister chromatids apart, then they're going to be separated into opposite parts of the cell, like so. Like before. Okay, so the spindle fibers contract, split the centromere, pull the sister chromatids to opposite poles, which leaves us with Telophase Two, where you'll have now 4 individual nuclear membranes form, each round, and I apologize if I don't get the combinations quite correct, I'm just remembering them off my top of my head. Okay, each with one of each of the chromosomes in it, like so. So we've now got haploid chromosomes, and the last stage, which will be cytokinesis, will literally mean that the cells will now separate, become 4 individual cells, either held together in the case of a plant cell or completely apart from each other in the case of an animal cell. But each one, as you can see, is genetically different to the next one and has half the number of chromosomes of the original parent. So there are 3 ways in which we introduce variation. The first way was crossing over. The second way was independent assortment. And the third way, which we haven't really discussed, is the fact that any sperm can join with any egg, what we call random fertilization, which means you get another whole load of variation introduced. If we go back up to the first diagram that we had of what is happening in meiosis, I'll scroll all the way back up. Here we go. We now know that in stage 1 of meiosis, the homologous chromosomes separate, homologous pairs separate, whereas in meiosis 2, the sister chromatids separate. So meiosis 2 is basically the same as mitosis in terms of what actually happens. I hope that's a bit more helpful. And that's the end of meiosis.